Good day, everyone, and welcome to this lecture video on cell division, particularly on mitosis and meiosis. So, the first part of this lesson is what we have already covered over October, which is the cell cycle, right? And its regulatory checkpoints. For this part onwards, we will talk about the two types of cell division that happens in human beings, and that is your mitosis and meiosis. So, this video is just the first part. We will just talk about mitosis here, while the second part will be about meiosis. Okay? So, for this lesson, we want you to first explain the processes involved in the phases of mitosis. And then second, to explain the processes involved in meiosis. At the same time, we want you to differentiate mitosis and meiosis, and then differentiate then the different types of meiosis in human beings, which is the formation of your sperm and your egg. Okay? But for the first part, we will need to go with number 1. No? We will talk about mitosis here. Okay? So let's recall the three types of cell division. So we have binary fission, which often happens in unicellular organisms, including some protists and all prokaryotes, right? Remember, these are the bacteria, archaea, that live as a single cell. Thus, cell division happens for reproduction purposes, no? Particularly asexual reproduction. Mitosis happens in multicellular organisms, no? Including eukaryotes like human beings. And this happens usually in somatic cells. When we say somatic cells, these are the body cells, no? Na hindi sex cells, okay? So, ang halimbawa nun, cells sa utak, cells sa kamay, cells sa mata, cells sa iba't ibang bahagi ng katawan, puwera dun sa mga parts na gumagawa ng inyong sperm or egg, okay? Now, what happens in mitosis is that it's a division wherein you're not making a new organism, but you're making a part of yourself, right? Unlike in binary fission, you're making a different organism already, no, a different individual organism. For mitosis, you're just replenishing the tissues that you have in the body, right? So the daughter cells here will have the same number of chromosomes as their parent cell. Okay? So, kung nanggaling yan sa lungs mo, dapat ang mabubuong cell din ay kamuka din nung pinanggalingan niya. Diba? The same is true for the cells in the stomach, in the eyes, in your cheeks, and all the other parts. Okay? All the cells that would be coming from them should be having the same DNA as their parents. No? So, ayan siya. And then we have meiosis, which is the division in sex cells. Okay? Whose daughter cells have half the number of chromosomes as the parent cell. Recall, in humans, we have 46 chromosomes. No? For normal human beings, 46 ang number of chromosomes natin. So, kung ang chick cell mo may 46 chromosomes at nag-divide siya, dapat yung dalawang cells na nanggaling sa chick cell, dapat ay chick cell din, ay mayroon ding 46 chromosomes. Diba? As for meiosis, dapat mga lahati. Diba? Na kung ang pinanggalingang cell ng sperm ay 46 ang chromosomes, ilan dapat ang magiging number of chromosomes ng sperm cell? Okay? It has to be half of 46. So 46 divided by 2 will give you 23. Okay? The same is true for the egg cell. The egg cell should have 23 as well. No? At bakit? Diba? Ba't kailangan mga lahati? No? Balikan. Ano bang purpose ng sex cells na to? Diba? They are used for sexual reproduction. No? That's why when a sperm having 23 chromosomes penetrated an egg cell with 23 chromosomes, the resulting cell is a zygote. Okay? Ikaw na yon, yung bagong nilalang, which will have 23 plus 23 chromosomes, and that is 46 chromosomes. Okay? So I hope you can still recall that. No? Now, and let's recall that mitosis is again the cell division in eukaryotic somatic cells. And when we say somatic cells, basta hindi sex cells. No? Kahit anong bahagi ng katawan, puwera sa sperm at sa egg cells. Okay? Ayan. So, shown on the right is the illustration or the GIF or the GIF showing you the process of mitosis that happens in somatic cells, no? So nakikita natin yung chromosome alignment and then yung hihiwalay dun sa mga sister chromatids, no? Para yung parehong cell ay magkaroon ng 46 chromosomes each. Okay? Ayan. Then meiosis happens just for the formation of your sperm and egg cells. Okay? So, let's define or recall the differences between your somatic and sex cells, diba? So, in terms of the examples, basically, pag somatic, it's just any body part, basta hindi sex cell, no? And the number of chromosomes is termed as, or it's called as diploid, no? 2N, okay? Ibig sabihin yan, okay, may dalawa kang N. Yung isang N ang galing kay father, yung isang N galing kay mother. So that's why you have 2N chromosomes, no? For the case of human beings, we have 46 chromosomes, no? For other organisms, they can have 10, they can have 16, they can have 32, they can have any number, no, any even number, no, for their diploid stage, no? But for sex cells, they are considered as haploid, no? So that's why it's only represented by N. Okay? Kalahati lang siya ng inyong diploid form. Okay? So for sex cells, they're considered haploid, so thus your sperm and egg should be half of the diploid stage, and that is 46 divided by 2, which is 23. Okay? Now, the cell division type that happens in somatic cells is always mitosis. For the formation of sex cells, it has to be meiosis, no? And we will talk more about genetic recombination when we talk about meiosis. So this does not happen in somatic cells. While in sex cells, they happen. Iba ang itsura, iba ang DNA ng inyong sperm cells compared dun sa inyong pinanggalingan. Una na nga, nangalahati na nga diba? Yung kanyang DNA, no? So, dun pa lang, alam mo nang naiba na. Diba? But for somatic cells, kung anong itsura ng pinanggalingang cell, yun din dapat ang itsura nung mga mabubuong cell. Okay? So, hindi pwedeng maiba yung itsura. Diba alam na natin anong mangyayari pag naiba yung itsura ng DNA? Nagiging cancer siya, diba? So, at yun yung ayaw natin pag nagmamitosis tayo. Dapat, kung anong ina, siya ring anak. Diba? Kung ano ang kalidad ng DNA sa pinanggalingan, yun din ang DNA na meron sa mga lalabas, no? Sa mga lilitaw from cell division, okay, or mitosis, no? Ayan. And then let's recall the chromosomes, no? Ano nga ba ulit yung mga chromosomes na to, di ba? Maraming nalilito tungkol sa differences ng chromosomes at DNA. Basically, ang chromosomes ay bahagi ng DNA, no? Kumbaga, ang DNA ng tao, yun yung naglalaman ng lahat ng identity mo. Diba? It's made up of nucleotides. Diba? Yung narinig nyo na yan nung grade 10 kayo. Diba? Yung adenine, guanine, cytosine, thymine, that AGCT, that's the entire code that makes up you. Right? So, kung ano man yung code na yun yung magdidikta kung sino ka. At dapat, kung ano yung code na yun, it should be shared by all of the cells in your body. Kasi pag may naiba lang na isang letra dyan, potentially, it could lead to mutations that can be a reason for cancer. Okay? So, let's recall again what are these chromosomes, no? Again, ang mga chromosomes ay parang segments lang din yan ng DNA. No? Hinati-hati yan into 46 parts, 'di ba? Kung 46 ang chromosomes ng tao, so meron kang DNA, hinati mo siya ng 46 times, 'di ba? At kung ano yung mga pulupot na magagawa mula dun sa mga pagkakahati-hati na yun yung mga chromosomes na lilitaw, di ba? So these are thread-like structures that are made up of protein and a single molecule of DNA, okay? That serve to carry all the genomic information from cell to cell. Kasi yun din yung mamanahin ng susunod na cell. Dapat ikaw pa rin yun, di ba? So it's found in the nucleus of your eukaryotic cells. No? Only eukaryotes will have nucleus at sa loob ng nucleus, doon mo lang makikita yung mga chromosomes. No? So, bakit may protein, sir? So, ang protein ay para lang mapulupot mo yung DNA. No? Kasi, syempre, knowing ang human DNA ay 2 meters no? Pagka ini-stretch out mo siya. And just imagine, lilit mga cells mo, di ba? So, ilang kilometrong DNA ang ang magagawa mo from all of the cells in your body. So para mapulupot siya sa loob ng inyong mga nucleus na nasa loob ng mga cells, na nasa loob ng katawan mo, 'Di ba? Kinakailangan mo ng mga proteins na ito. Okay? So, yun yung no? Parang nakaspool, nakapaikot yung mga DNA para hindi sila masyado magiging maluwag or talagang lalagpas sa size ng cell, no? Para mapagsiksikan natin sila, 'di ba? Yan. So, a normal human being has 46 chromosomes in each of their cells. Now, the question is, after one round of the cell cycle in mitosis, how many chromosomes do each daughter cell get? And as we recall, in mitosis, diba, pagka okay na isang round of mitosis or cell cycle natin, dapat ang magiging anak mo na cells, kamukhang-kamukha niya dapat yung pinanggalingan niyang ina. Diba? So, kung 46 ang number of chromosomes ng magulang, dapat 46 din yung makukuha ng anak. Okay? Okay? Now, meron pa tayong isa pang term noon, tawag ay sister chromatids. Diba? May chromosome na nga may chromatids pa. No? So, ano ba tong chromatid na yan? Diba? So, sister chromatids yung usual na tawag sa kanya kasi nga, magkakasama yung kopya yan ng chromosome. No? So, this is the two identical copies of a chromosome that is ideally formed after S phase. No? Balikan ng cell cycle. Inaral natin sa cell cycle na meron siyang S phase na kung saan doon magduduplicate yung DNA. So, pag na-duplicate yung DNA, di maduduplicate din yung mga chromosomes mo. Diba? So, nung dinuplicate mo yung chromosomes nung time na yon, di magiging, kung isa lang siya nung una, magiging dalawa na. At magkadikit sila. Kaya ang tawag sa kanila ay mga sister chromatids. No? Just imagine kung 46 ang chromosomes mo na ganito, edi magkakaroon ka ng 92 chromatids pagkatapos ng S phase. Diba? Ito ay isa lang chromosome. Pero meron ka pang 46 na ganito. Okay? 46 na ganito sa loob ng inyong mga cells, no? After ng S phase. Diba? So, balikan. If humans have 46 chromosomes in their cells, how many chromatids do they have at G2 phase? So G2, nangyari siya after ng S phase. So nagduplicate na yung DNA. Dumoble na yung chromatids ng inyong mga chromosomes. So dapat, meron na siyang 92 chromatids. Okay? Pag ang tanong, how many chromatids do they have at G1 phase? At G1 phase, ilan ang kanyang chromatids? Okay? Wala pang cell division or wala pang synthesis phase, hindi pa nagduduplicate yung DNA. Thus, the number of chromatids will remain at 46. Okay? Magiging 92 lang kapag okay na yung S phase. No? Ayan. So, sana klaro yun. No? Dumodoble siya, di ba? Kasi nga gusto mong paghiwalayin ulit sa mga bagong cells sa susunod na division. Okay? Ayan. So, may mga iba't iba pa tayong mga parts ng ating mga chromosome. So, una na dyan, yung inyong centromere. Okay? So, yung centromere is just the central region of the sister chromatids in a chromosome. So, dyan sila nagkakadikitan. Diba? It's just the central region of your chromosome. No? Magkaiba siya sa centrosome. Okay? Ang centrosome ay isang organelle na siyang magiging dahilan kung bakit magkakaroon ng mga spindle fibers during mitosis. Okay? So, centrosome is the organelle. Centromere is just the part of your chromosome. Okay? So, wag malilito dun sa dalawa. No? Ayan. Okay? And then, finally, may binabanggit tayong kinetochore. At isa siyang region dun sa may centromere na kung saan dun didikit yung mga spindle fibers na ginawa ng centrosome. Okay? So, remember, in the M checkpoint or the mitotic checkpoint, ang gustong tingnan ni na cyclin at CDK doon ay kung naka-attach na ba yung mga spindle fibers sa mga chromosomes. Okay? Doon sa mga chromatids. So, kung saan sila naka-attach sila tinatawag nating kinetochore. Okay? Balikan. Why will the cyclin and CDK check the spindle fiber attachment to chromosomes at the mitotic checkpoint? Ba't kailangan naka-attach? Pwede namang hindi. Diba? Ano mangyayari kapag hindi mo in-attach ang mga chromosomes, mga spindle fibers? Okay? Take note, anong magiging role nila later? Ang mga spindle fibers, sila yung maghahatak dun sa mga chromosomes sa gitna para madala sila sa dalawang bahagi nung magdi-divide na cell. Diba? So, kung walang spindle fiber, may iwan lang sa gitna yung chromosome mo. Saan yung dadalhin, 'di ba? So, hindi magkakaroon ng equal division kung merong may iwan sa gitna. So, that's why in the mitotic checkpoint, tinitingnan nila cyclin at CDK kung nakaayos na ba yung attachment na yan sa mga chromosomes. No? Otherwise, makukulangan ka ng chromosome sa isang cell. Diba? Pag may mga naiwan pa sa gitna. Diba? Yan. So, balikan din natin yon, no? Ayan. In the exam naman kasi, what I want you to know of is the process, no? And the why, no? Bakit ganito? Bakit ganyan? Ba't kailangan mangyari yon? Kung hindi yan mangyari, anong pwedeng mangyari? Diba? So, mga ganong klaseng tanungan yung i-expect natin sa exam. Okay? So, ayan. Okay? And let's recall what are the functions of cell division. And as we recall, no, para saan ba siya? Bakit kailangan mag-divide ng mga cells natin, no? So, ulitin, no, it's for reproduction, particularly in unicellular organisms, no? Through binary fission, talaga dumadami yung mga bacteria, yung mga protists, di ba? And for human beings, for multicellular entities, it's for growth and tissue renewal, di ba? For the repair of damaged tissues in the body. So, syempre, pagka nasira yung mga cells na yan, kailangan silang punan, no? Kung hindi mo sila mapupunan, talagang maiiwan na parang airspaces sa loob ng katawan mo, di ba? Magiging, ang laman mo, puro hangin na lang, no? Kung walang mitosis, kung walang mga cell division na yan. Okay? Ayan. And let's recall the cell cycle, okay? The cell cycle is composed of the interphase and the mitotic stages, no? What we will be discussing for this part is just this small part of mitosis, no? For the bulk of the cell cycle, it's just doing nothing but growing and synthesizing its DNA, diba? Or duplicating its DNA. In the interphase stages, parang walang nangyayari sa cell, kundi lumalaki lang siya, diba? Pero yung di-discuss natin ngayon ay yung maliit na bulto ng cell cycle at yan yung mitosis, no? It's just a part of the whole cell cycle. And this is the phase, no, of division, okay? Magdi-divide yung mga cells mo, no? So that, you know, from one cell, there will be new cells that will be produced, okay? So let's now begin explaining the processes involved in mitosis, okay? Simula pa lang ng lesson, di ba? So anong nga ba mitosis, no? Again, it happens only in somatic cells. It's the division of cells that are somatic in nature, body cells. Mga cells mo sa mata, cells mo sa panga, cells mo sa talampakan, cells mo sa atay, sa bato, sa baga mo, mitosis ang nangyayari sa lahat ng yan, no? At yung mga number of chromosomes ng mga daughter cells na mabubuo from mitosis, katumbas din nila yung number of chromosomes nung pinagmulan nilang magulang na cell. Okay? So, kung may 46 chromosomes sa pinagmulan, dapat lahat ng anak na cell ay magkakaroon din ng tig for 46 chromosomes. Okay? So, yan. So, let's see what happens here in the diagram, no? So, as we can see, okay, so merong mga decondensing or condensing of the chromosomes that are in red. Okay, nakita niyo 'yon, no? The red ones are the chromosomes that are condensing, no? And then later on they will be aligning in the middle bago sila mapaghiwalay patungo sa magkabilang panig, no? Ano naman yung green, 'di ba? So the green ones are actually your spindle fibers. Okay? So these are the spindle fibers that would be generated from your centrosomes. So yung mga bright green sa gilid, centrosomes 'yun, no? Yun yung origin ng mga spindle fibers. At sila yung maghahatak dun sa mga chromosomes patungo sa magkabilang dalawang panig, 'di ba? At yung dalawang panig na 'yan, magiging separate cells din 'yan ultimately. Okay? So I hope you're observing the process. You can imagine what's happening, no? This is a real footage with stained cells na kung saan ang stains sa mga chromosomes ay red. Tapos ang ginamit sa mga spindle fibers ay green. Okay? And then they are fluorescent. So ibig sabihin, kapag sininagan mo ng UV light, yan yung i-reflect nilang light pabalik sa mga mata natin. So kaya ganyan yung kulay nila. Okay? So, mitosis comes in 4 or 5 phases. No? Meron pang pro-meta phase, diba? Pero narinig nyo na to. Grade 8 pa lang, dinidiscuss na ang mitosis sa atin. So, we have the prophase, metaphase, anaphase, and telophase. Okay? So, PMAT lang yan, class. No? PMAT. Diba? Prophase, metaphase, anaphase, telophase. No? Yung pro-metaphase, feeling ko hindi nyo naman kailangan talagang tandaan at all. No? It's just there to show you the transition between the prophase and the metaphase stages. Okay? So, let's recall what happens to the cell before mitosis. Again, it's just in the interphase. Okay? So, kung galing siya sa interphase, dapat buo pa yung nucleus mo. So, kung buo yung nucleus, it must have an intact nuclear membrane. Okay? At the same time, it should have a distinct nucleolus inside the nucleus. So as you can see in the diagram, this is the cross-section of onion tip, no, of an onion root tip. Okay? So yung ugat ng onion, pinatag mong ganon, tapos nilagyan mo ng stain, ito na yung nakikita mo. No? So yung mga bilog-bilog na nakikita ninyo, okay, those are your nucleus. At yung dark na bilog sa gitna, o sa loob ng mismong mga bilog na yan, yun yung nucleolus. Okay? Bahagi din siya ng nucleus. Ano nga ba yung nucleolus ulit? No? So, ito yung region ng nucleus na siyang nagpo-produce ng mga ribosomes. Diba? Ayan. And as you can see, wala pa kayo nakikitang chromosomes. Hindi nyo pa kayang bilangin. Kasi nakapulupot pa yung DNA dyan sa loob ng inyong mga nuclei. Okay? So, yan ang itsura niya, no, at interphase, no, just before mitosis. Okay? So we'll now go to each of the phases, no? As you can see, itong pointed na cell dun sa diagram or in the microphotograph on the right, that is already the cell that is undergoing metaphase. Okay? Metaphase na yan. Ibig sabihin, nasa kalagitnaan na siya ng mitosis. Diba? Ibang-ibang itsura ng cell kumpara sa iba pang mga cells na nasa interface. Ano? So that's why, kapansin-pansin talaga sa microscope kapag nag-undergo ng mitosis ang mga cells. Okay? Ayan. So, let's now go to the different stages of your mitosis. Diba? The first phase is the prophase. No? And take note, ano bang gusto mo mangyari dyan, di ba? So, una, buo pa yung nucleus mo. Kailangan nga paghiwalayin yun, di ba? Yung mga DNA na nasa loob niya, no? So, ano dapat mangyari? Mabuwag yung nucleus na yun. So, you have to expose your DNA into the cytoplasm. Your DNA here is now in the form of chromosomes. So, they will begin condensing. Madali mo na sila makikita, no? Especially with stains, no, that you will put on the cell or the tissue line, di ba? Yan. So from there, the spindle fibers will begin to emerge from the centrosomes. Again, these are the centrosomes, the organelles that will make all of these spindle fibers, no, patungo sa mga chromosomes, no? Ayan. Then the nuclear envelope will break down and the nucleolus will disappear. Bakit? Ba't kailangan buwagin? No? Again, ang goal mo ay mapaghiwalay mo ngayon DNA, di ba? Hindi mo sila mapaghiwalay kung nakakulong pa sila sa loob ng nucleus. So, kailangan mabuwag yon para mailabas mo yung mga chromosomes at para mas madali mo na sila mapaghiwalay. Okay? So, ganun dapat ang prophase, di ba? And, ganun lang kasimple yon, no? So, in pro-metaphase, which I don't think you have to really remember at all, no? It's just a transition phase between your prophase and metaphase. Okay? So, ano nangyayari dito? Your kinetochores will begin to appear at the centromeres. O, ano ulit mga yun? No? Ano yung mga chromosomes natin? Di ba? Meron silang bahagi sa gitna na kung tawagin ay centromere. At mula sa centromere, lilitaw yung kinetochores. Para saan ulit yon? Ito yung attachment region ng mga spindle fibers para mahahatag sila sa magkabilang panig ng cell. Diba? At the same time, no, your mitotic spindle microtubules will begin attaching to kinetochores. O bakit? Again, para mapaghiwalay mo sila later on. Sila yung hahatak, diba? And centrosomes move toward opposite poles. No? So, centrosomes, sila yung gumagawa ng spindle fibers. So, kung ito yung cell, yung mga centrosomes mo, nasa gilid sila, gumigilid sila. Hindi sila pwedeng nasa gitna, di ba? So, kung gusto mo mabatak yung cell at para mapaghiwalay mo into two new cells, di ba dapat nasa magkabilang gilid? Di ba? So, yun naman yung no? Common sense naman siya, di ba? So, ayan na, pagka tinanong kayo bakit kailangan pag-isip-isip natin, bakit nga ba, di ba? Pwede naman sa gitna. Pwedeng isa sa taas, isa sa gitna. Pero bakit magkabilang gilid? Okay? So, by means of common sense, di ba, you want to divide the cell, thus, para mabatak mo talaga, di ba? So, they have to be at the opposite poles, no, in your cell. Okay? So, that happens at pro-metaphase. Okay? Now, in metaphase, the mitotic spindle is fully developed. No? Yung mga spindle fibers, they are now at their longest at this time. No? The centrosomes are now at the opposite poles of the cell. No? Nung una kasi wala pa sa gitna yan, nung pro-metaphase, nagpupunta na sila sa mga gilid. Diba? At the same time, your chromosomes will begin aligning in the middle. And that alignment, no, is called that imaginary line called your metaphase plate. Okay? Yung metaphase plate, hindi naman talaga siya totoong region, no? It's just an imaginary area where all your chromosomes, no, will be aligning in the middle, no? At yung bawat chromosome na yan, may mga sister chromatids sila. 'Di ba? Yung isang sister chromatid na connect siya sa saan? Sa spindle fiber ng isang pole. Habang yung kabilang sister chromatid na kabit naman siya sa spindle fiber ng kabilang pole. Okay? So, para mapaghiwalay mo at maging separate chromosomes completely. Diba? So, again, bakit kailangan mangyari yon? No? Bakit kailangan magkabilaan? Para maputol mo at mapaghiwalay mo eventually yung mga sister chromatids, which will become the future chromosomes. Okay? So, that happens during the metaphase stage. No? And let's recall, during the metaphase stage, dyan din nangyayari yung mitotic checkpoint. Tataas ang concentration in acylin at CDK during this time because they want to monitor if the chromosomes are attached to all spindle fibers. Otherwise, they have to be corrected. No? Hindi pwedeng may maiwan sa gitna kasi mababaliwala yung cell division. Diba? Hindi ka makakabuo ng cells na may equal number of chromosomes, no? Kung meron mga may iwan sa gitna. Okay? Yan. Now, I'd like to add here that some spindle fibers do not attach to the sister chromatids, no? They go further towards the opposite pole. Okay? So, ang tawag natin doon ay yung mga non-kinetochore microtubules. Kasi nga, hindi sila nakakabit sa kinetochores. Diba? Ang kinetochores, nandun siya sa mga chromosomes, no? Dun sa mga nasa centromere ng mga chromosomes natin. Okay? Ayan siya, no? And let's recall saan nangyari yung spindle fibers. If we recall our lesson on cytoskeletal elements, they are made up of these dynamic microtubules, no? Either humahaba sila o umiikse. Diba? So, they're dynamic. Pabago-bago yung sizes nila. No? So, ayan. So, para saan yung mga non-kinetochore microtubules, no? Ayan. Okay? So, sabi rito, no, during the anaphase, your non-kinetochore spindle fibers or the non-kinetochore microtubules will lengthen. Sila yung humahaba habang yung mga kinetochore spindle fibers umiikse. No? So, in anaphase, okay, what will happen then? Okay? So, you have here your cohesin proteins which will begin breaking down. No? Pag nag-breakdown na sila, mapaghihiwalay mo na yung mga sister chromatids. So, they will now become chromosomes na ng magkabilang cell. Diba? And they're now pulled towards the opposite poles. So, with that being said, your kinetochore spindle fibers will shorten. Umiiksi sila. Diba? Kasi kailangang hatakin sa magkabilang gilid. Habang ang mga non-kinetochore spindle fibers, sila yung humahaba. Okay? And for what reason? No? They have to elongate the cell. Bakit kailangan i-elongate? Again, ano bang proseso to? Cell division. The more you extend the size of the cell, mas lumalaki ang chance na maghihiwalay na sila later on. Okay? Diba gano'n naman? Pag malayo na kayo sa isa't isa, naghihiwalay na kayo. LDR, diba? Pero parang gano'n yung nangyayari. Diba? So, yan siya, class. No? So, in anaphase, diba, mula sa gitna, pinaghiwalay na, no? Naghihiwalay na. Nagpupunta na sa magkabilang panig ang mga chromosomes natin, no? Mula sa mga dating mag-sister chromatids. Okay? Yan. So let's recall again, the kinetochore spindle fibers will shorten because you need to pull the chromosomes toward each pole, right? While you have your non-kinetochore spindle fibers that will lengthen. And that is again to elongate the cell for easier separation, no? Para mapadali mo yung paghiwalay nila. Kasi in the end, gusto mo ng bagong cell, 'di ba? So, pahabain mo ng pahabain. 'Di ba? Ayan. Okay? And finally, we have the telophase. And telophase is simply the complete opposite of what happens in the prophase stage. No? Chromosomes here arrive now to the opposite poles and they begin decondensing. No? Unti-unti na silang lalabo sa paningin mo. Malulusaw na sila or magiging long strand na sila ng DNA ulit. So, wala na yung... Bali, doon pa rin yung chromosomes kasi DNA pa rin sila, pero hindi mo na sila makikita. Diba? Eventually, your nuclear envelope material will begin to develop, and they will surround each set of chromosomes. So, in this cell, okay, during telophase, ilan ang nuclear membrane na mabubuo mo? Okay? Di pwedeng isa. Diba? Kasi kung isa, ano yun? Hindi ka nagdi-divide, diba? Dapat dalawa na. Kasi nasa dalawang magkabilang panig, dalawang nuclear membrane yung mabubuo mo. Okay? In prophase, binuwag mo yung isang nuclear membrane. In telophase, may mabubuo ka ng dalawang nuclear membrane or nuclear envelope. Okay? Eventually, you'll form your nucleus. Nandun na yung nucleolus, buo na siya ulit, ready na siya to go to the interphase after division. Diba? The mitotic spindle breaks down also during telophase. Bakit? No? Kailangan mo pa ba niyan? Hindi na, kasi natapos na yung trabaho niya, diba? Ayan, again, common sense naman din siya, no? So, yan, pagka may mga lumalabas na tanong sa exam na bakit, no? Minsan, baka pwedeng common sense na lang yung panggalingan ng sagot, diba? So, bakit kailangan madurog ng mitotic spindle? No? Kasi, mitosis is almost over. It's no longer needed because it has done its job already. Tapos na yung trabaho niya. Nadala niya na yung chromosome sa magkabilang panig. Okay na rin yung size ng cell, pwede na siyang maghiwalay. Diba? Okay? So, ultimately, this will lead to cytokinesis, no? Which is a different stage after mitosis, okay? So, to be clear to everyone, mitosis is simply the division of the nuclear components. That includes the DNA, yung mga chromosomes na yan, yung paghiwalay nila, yun. Okay? Yun. No? Pero yung paghiwalay ng cell membrane para magkaroon ka ng dalawang cells, ang tawag natin sa proseso na yun ay cytokinesis. At mauuna ang mitosis bago ang cytokinesis. Kailangan mapaghiwalay mo muna yung DNA bago mo paghiwalayin yung cell. Diba? Kasi kung pinaghiwalay mo muna yung cell bago yung DNA, edi yung isang cell walang DNA. Diba? Wala rin. Mamamatay din yun. No? So again, common sense lang din. Alin mauuna dun sa dalawa. Diba? So that division of the cell membrane is called cytokinesis and it happens after telophase. Pag okay na yung nucleus, na may dalawa ka ng nuclear membranes, okay na yung DNA, nag-decondense na sila, bumalik na ulit sa normal, edi paghiwalay mo na yung cell. Diba? So after the cytokinesis, the cell is ready to face another round in the cell cycle. Okay? So another cell cycle ulit yun, tapos mag-mitosis ulit. Diba? Pero yung cytokinesis ay naiiba sa parehong animal and plant cells. Paano? No? So we have here your cleavage furrow that will form between two separating cells in animals. Take note, in animals, they do not have cell walls. They just have this immediate outer covering of the cell membrane. Thus, pag nag-divide kayo niyan, talaga makikita mo na merong puwang sa gilid nila, no? Na ang tawag natin ng cleavage, di ba? So that's the sign that the cell is already entering cytokinesis. Maghihiwalay na yun eventually. Di ba? But in plant cells that have cell walls, hindi mo makikita yung cleavage furrow. Instead, what you will see is a cell plate. That cell plate will develop with the action of the Golgi bodies to form between two new cells. Okay? Kumbaga, may bago kang cell wall na galing sa action ng inyong Golgi bodies. Okay? So, yun lang naman pinagkaiba ng animal cells at plant cells. Basically, it's because also of their structural differences. Animal cells do not have a cell wall, while plant cells have it. Okay? Yan. And now let's have mitotic index. Now, you have encountered this already in the Space Lab 3, where you were given a material showing you the effects of P53 into different cells, no, in terms of their mitotic index. Pag mas mataas ang mitotic index, ibig sabihin, mas maraming cells yung nag-undergo ng mitosis. Pag mas mababa, mas konti o kulang yung mga cells na nag-undergo ng mitosis. Diba? So, it's simply the frequency of the cells that are undergoing mitosis given a whole number of cells. No? So, how do you calculate for the mitotic index? No? It's calculated as the number of cells undergoing mitosis divided by the total cells present. Okay? Dalawa lang yan, class. Diba? Nagmamitosis o hindi mitosis. So, kung hindi mitosis, interface yon. Yun yung buo pa yung nucleus, buo pa yung nucleolus sa gitna, wala pang condensation of the chromosomes. So, yun na yun. Diba? Ayan. So, given this illustration or microphotograph on the right, what is the mitotic index of the view of onion cells here? So you can simply calculate by counting all the distinct nuclei or the distinct cells that are undergoing mitosis. So, of course, you have to count first ilan yung nag-undergo ng mitosis. So in this figure, you'll see that there's one, yung pointed one, diba? Well, for the rest, they are 11. Okay? So, these are the cells that are undergoing interphase. No? So, how do you calculate it then? So, mitotic index is simply 1 divided by 11 o 12. Okay? It has to be 12. Total nga, di ba? Kasama din yung nagmamitosis. Okay? Interphase plus mitosis. No? Okay? So, that's 1 over 12, which is actually 0.083 or 8.33%. Okay? So, ganun lang kadali yung pagka-calculate ng mitotic index. Na magbibilang ka lang naman, di ba? Ayan. So, in your exam, you'll see some examples showing you the mitotic index or perhaps the number of cells that are undergoing mitosis. No? So, may mga ganun tayong mangyayari sa exam. But don't worry, you don't need to calculate anymore because all you need to do is to interpret what you're seeing. Di ba? So, yun lang naman siya. Okay? Para saan yung mitotic index? No? So, it is being used no to study cancer cells. Kasi mga cancer cells, diba, usually, hindi controlled yung kanilang cell cycle. So, that's why I have this discussion prompt with you. No? When cyclin fails to monitor DNA quality, the cell may proceed with the cell cycle even with erroneous DNA, which may lead to cancer. Diba? Now, let's try to compare normal and cancer tissues. Which tissue will have a higher mitotic index? And why? Okay? So, pag-isipang maigi. No? Take note. Kapag cancer cell ka, walang nagko-control sa cell cycle mo. So, ibig sabihin, anytime pwede ka mag-multiply. No? Pwede ka mag-divide. Kahit mali-mali yung DNA mo. So, what you'll see here is that cancer tissues will potentially have a higher mitotic index. Okay? Kasi wala nang nagko-control sa cell cycle nila. Walang cyclin, walang CDK na mag-aalaw na, oops, wag ka muna mag-divide kasi hindi ka pa okay. No? Eh dahil sira si cyclin at CDK, wala nagmamando sa cell cycle. So, talagang dire-diretso yung pagdi-divide ng mga cancer cells. No? That's why, habang tumatagal, pag hindi naagapan ng cancer cells na yan, nagiging tumor sila. At eventually, malaki yung magiging epekto nila sa ating katawan. No? So, ayan. I hope you have the same answer as I did. Okay? Na sana na-figure out niyo siya bago niyo pinakinggan yung sagot ko. Okay? Oh, how about this one? You made a drug that stops the cell cycle in cancer cells, but not in normal cells. Assuming nahanap mo na ang solusyon sa cancer, which should show proof that you did it correctly. Oh. So, using the concept of mitotic index, ano dapat yung mangyayari kung gagawa ka ng experiment? Okay? So, A. After treatments, the cancer cells have lower mitotic index compared to normal cells. B. After treatment, cancer cells have equal mitotic index with normal cells. Or C, after treatment, cancer cells will have higher mitotic index than normal cells. Or letter D, after treatment, both cancer and normal cells, okay, will have lower mitotic index. No, parehong bababa. Yan. So, ano sagot? Pag-isipan. Ang gusto mo ay i-stop mo yung cell cycle ng mga cancer cells. So, pag tinigil mo yung cell cycle, ano mangyayari sa mitosis? Mangyayari pa ba? Dapat hindi na. Okay? Pero syempre, ang condition mo rin dito, dapat hindi mo rin pigilan yung mga normal cells. No? Kasi kung pinigilan mo yung mga normal cells, parang lason yan, mamamatay ka. 'Di ba? Solusyon nga sa cancer ang gusto mo, 'di ba? So, dapat ang target mo lang, cancer cells. No? So, the right answer, okay, should be letter A. Okay? After treatment, the cancer cells will have a lower mitotic index compared to your normal cells. Diba? So, ibig sabihin nun, na-slow down mo na yung pagpaparami ng mga cancer cells. No? At solusyon siya sa cancer. Diba? Kung ayaw mong mag-progress yung inyong tumor, diba? You want to prevent it from happening. So thus, you need to resolve it using the idea of stopping the cell cycle in cancer cells. Okay? Ayan. Now, let's have item number 2. Using the same situation, can the drug kill the cancer cells? A, yes, it can induce apoptosis in cancer cells, but not in normal cells. B, no, stopping the cell cycle will kill the normal cells and not cancer cells. Or C, yes, when the cell cycle is stopped, the cancer cells should die. Okay? Or D, no, it can only reduce the cell ano yun? Sorry, nakaharang yun. Yeah, it can only reduce the cell division to prevent further tumor enlargement. Ayan. Okay? Have you given it a thought? So, for this case, the right answer is letter D. No, it can only reduce the cell division to prevent further tumor enlargement. Again, what you're trying to do is to only stop the cell cycle, but not ask the cell to undergo apoptosis. Again, apoptosis is programmed cell death, no? Hindi yun yung role ng drug na na-discover mo. Some drugs would do that, but some drugs will only inhibit the cell cycle. Okay? So these are different things. So you have to analyze carefully when you're given this kind of situation. So what you're seeing here is just a solution that will stop the tumor enlargement. Okay? Pero yung cancer, nandun pa rin. So that cancer may require a different solution. Okay? Either surgery, radiotherapy, or chemotherapy. Diba? So may ibang droga para sa pagpapalaki ng tumor, para maiwasan yon. At may ibang droga na nakakapag-resolve ng mismong cancer cells. No? But they really come at a cost, and with most of them having side effects. No? That's why right now we're still in the hunt for the right cure to cancer. Okay? So, ayan. I hope that you appreciate the concepts of mitotic index and how is it being applied in cancer research. No? So, ayan. Okay? And that ends our lecture, the first part of the lecture for mitosis. And I hope you learned something new and apply it in your exams this week. Good luck.